In the previous video, we had finished talking about the prokaryotic cell wall, specifically the bacterial cell wall. We addressed the gram stain, and we saw how the gram staining procedure can help to differentiate between gram-positive bacterial cells that have that thick peptidoglycan layer in their cell wall versus the gram-negative bacterial cells that have a more complicated bacterial cell wall, which has a thin layer of peptidoglycan just outside the plasma membrane, and then outside of that is that extra outer membrane that oftentimes can be toxic to the host organism. Okay, on top of that, let's talk a little bit about why in the world would it might it be important for, let's say, a doctor to figure out whether your infection that you have is either gram-negative or caused by gram-positive bacterial cells. So many of the antibiotics that we have currently developed actually attack the peptidoglycan and can damage those bacterial cell walls. Bacterial cells have that damaged cell wall, they don't have the protection, they might take up too much water, causing the cell to burst. A lot of things can happen when that cell wall is not intact. If an antibiotic attacks the peptidoglycan layer of the cell wall, right, for the gram-positive bacteria, it's pretty easy for that antibiotic to attack the peptidoglycan layer, degrade it, and now the gram-positive cell is at risk. Gram-negative cells, though, remember they have that outer membrane. Not only is this guy nasty because it can be toxic, but that outer extra membrane actually protects that thin layer of peptidoglycan from damage by antibiotics. So many of the gram-negative bacteria are actually antibiotic resistant if that antibiotic specifically targets that peptidoglycan layer of the cell wall. So if you guys are going to be picking, right, you have to have an infection. Are you going to want a gram-positive bacterial infection or gram-negative bacterial infection? My bet, my personal preference, I'd probably go for the gram-positive. Those guys don't have that extra toxic outer membrane as part of the cell wall, and they tend to be less antibiotic resistant, unlike the gram-negative cells, which can be antibiotic resistant depending on which antibiotic you're given. That one targets the peptidoglycan layer. The gram-negative cells probably won't respond to it. Okay, on top of that bacterial cell wall, some bacterial cells have an extra layer. This is called the capsule. And this capsule, if you take a look at this image right here, so here's our brown-orange bacterial cell, right? It's bound by the plasma membrane. On top of the plasma membrane, they have that bacterial cell wall, here shown sort of in yellow. And then just to the outside, there's sort of this fuzzy layer, right? This is called the bacterial capsule. It's usually made out of polysaccharides or proteins, and it can cover many prokaryotic cells. The main function of it is to help to stick to substances. So for example, this cell right here looks like it's causing an infection, and it's stuck itself to a tonsil cell of that particular host organism. It's that sticky capsule that helps the bacterial cell stick to that tonsil cell. Okay, some prokaryotic cells also have these thin, long projections, which are not going to be motile, but they're called fimbria, and they allow the cells to stick to each other and to also stick to the other substrates around them, very much like the bacterial capsule. The fimbria, this can help various cells within a bacterial colony to stick together so that they can actually grow as a colony. Okay, sometimes these fimbria, they're actually, a cell might have a longer, well, it looks like a fimbria, but it's actually not exactly a fimbria. We would call this a pili or a sex pili, pilus. These are usually longer, and as we will learn in one of the later chapters when we get to cell division, bacterial cells, prokaryotic cells, don't divide like eukaryotic cells do, and prokaryotic cells usually divide by a process called binary fission. Essentially duplicate the DNA, make two new daughter cells, and there's no mixing of genetic information. So every single cell that is a direct descendant from that original cell is going to be a clone, an exact copy. Sometimes mixing the genetic information is good, right? Maybe if some cells are resistant to antibiotics, others perhaps are not, it's good to mix some of that DNA, at least in terms of the life of that particular species of bacteria, so that the bacteria that weren't resistant to the antibiotic may now have the genetic information that they need to be resistant. So these sexpili allow prokaryotic cells to actually exchange some of their DNA, whereas their process of reproduction doesn't allow for that. Okay, some bacterial cells, some prokaryotic cells also have the ability to move. Right? They move by a specific mechanism, which we'll talk about in a second. But usually these bacterial cells are going to live in what we call a heterogeneous environment. So this environment might have different parts that have different molecules in it, for example. And bacterial cells will exhibit a phenomenon that's called taxis. Think taxi, right? To move. This is taxis. And more specifically, this is the ability to move towards or away from a specific stimulus. A more specific kind of taxis is called chemotaxis, and this is the movement either towards or away from a chemical stimulus, hence chemo for chemical and taxis for movement. When these bacterial cells, for example, move towards or away from a particular chemical signal, when they are exhibiting chemotaxis, they usually do so by propelling themselves forward using a flagella. Here's where bacterial cells and eukaryotic cells both have these structures called flagella, but they look different and they behave a little bit differently as well. So most moral bacteria will move themselves using a flagella. They can have one flagella and they have multiple flagella. Sometimes the flagella might be scattered on the surface of the bacterial cell, or maybe those flagella might be sort of concentrated at one end or another end of the cell. Right? This is different from those eukaryotic cells that have flagella. Usually, if a eukaryotic cell has a flagellum, it's only going to have one, right? That's it. Bacterial cells can actually have many. Okay. One of the last parts that I want to talk about in terms of prokaryotic cells, and hopefully you somewhere in the back of your head have sort of thought about this. When we talked about energy processing, right? Eukaryotic cells have mitochondria. Mitochondria are important for cellular respiration. Mitochondria make ATP. Chloroplasts, right? Those organelles perform photosynthesis, and they can produce sugars for a cell that can then be shuttled into the mitochondria to make ATP. Well, we know that prokaryotic cells don't have any membrane-bound organelles. That means that they don't have any mitochondria. They don't have any chloroplasts. They still need a source of energy, right? They still need to make energy-rich molecules to be able to pay for all sorts of cellular processes. So how do prokaryotic cells do this? They do have a certain amount of internal organization. So there are no membrane-bound organelles, right? Those don't exist. But some prokaryotes do have specialized membranes that can perform certain metabolic functions. And these metabolic functions are actually reminiscent of what we know so far about cellular respiration and photosynthesis. Usually, these specialized membranes are actually just an infolding of the plasma membrane itself. So here we have two examples of cells. The one on the left is an aerobic prokaryote. This is a prokaryote that needs oxygen in order to be able to utilize the energy that it gets from certain molecules. And so it'll have these infoldings of the plasma membrane, which are called respiratory membranes. And these are very similar to those membranes of the mitochondria, right? Except, again, it's not a separate membrane. This is just an infolding of the plasma membrane. On the right-hand side here, we have a photosynthetic prokaryote. And these guys have, again, infoldings in the plasma membrane, but these infoldings more so resemble those thylakoid membranes that we talked about in the chloroplast. So these two bacterial cells, the one with the respiratory membrane and the one with thylakoid membranes, again, just infoldings of the plasma membrane that have become a little bit more specialized. These guys were probably descendants from those ancient prokaryotic cells that either had the ability to do cellular respiration or had the ability to do photosynthesis that were absorbed or phagocytosed by that ancestor of a eukaryotic cell and eventually became either the mitochondria or the chloroplast. Again, going back to that endosymbiont theory. Okay, we still need to talk about prokaryotic DNA. So far, we know that all cells have DNA. We've talked about eukaryotic cells having multiple chromosomes. Something we may or may not have addressed yet is that eukaryotic chromosomes are linear, so one long DNA molecule. Prokaryotic chromosomes are circular, so they exist as a circle, no beginning, no end. And the prokaryotic genome has far less DNA. Usually, the genome consists of only one circular chromosome. Again, to reiterate, we know that that chromosome is not surrounded by a membrane, right? There's no nucleus in prokaryotic cells. Instead, this membrane is located in that region that we call the nucleoid. So one sort of segment of the cell, one region in the cell where the DNA sort of hangs out. All right, some species of bacteria also, on top of having their big old circular chromosome, they're also going to have smaller pieces of DNA that we call plasmids. These are really, really small circular pieces of DNA. And scientists have been able to exploit these to copy genes, many, many numbers of genes. For example, you can take a human gene. Let's say you're interested in a particular human gene, maybe hemoglobin, and you want to produce a lot of that gene, right? Or maybe insulin, a lot of that gene. You take that gene for that particular protein and you stick it into bacterial plasmid. You put that bacterial plasmid back into the bacterial cells. You provide nutrients for those bacterial cells, and what do they like to do? They like to divide. And as they divide, they automatically are going to replicate all their DNA, including those plasmids, making lots and lots of copies of that gene that you, perhaps as a scientist, are interested in. This is essentially what we as scientists call cloning, making multiple copies of a particular gene. All right, so to give you a closer look, here is the inside of a prokaryotic cell, and all of these little strands are part of the bacterial chromosome. It's circular, but it's all sort of bunched together in that nucleoid region of the cell. On the outside here, you can see these smaller, much, much smaller, circular pieces of DNA. Those are those plasmids that we were just talking about, those extra pieces of DNA. Okay, so to conclude, there are some structural differences between prokaryotic cells and eukaryotic cells we talked about. We also talked about those specialized membranes and also how differently the DNA is organized. There are plenty of other differences. These differences include differences in DNA transcription, DNA replication, and also translation. We're going to cover these differences as we get to all three of those processes individually. On top of that, this last point says that these differences between prokaryotic and eukaryotic cells are differences that we in medicine and science totally exploit. For example, we develop antibiotics that will target those specific differences so that when we administer an antibiotic, we want to only damage the bacterial cells. We want to leave all the eukaryotic cells of the human being totally and completely intact, right? No harm to the host, but we definitely want to kill the bacterial cells causing the infection. So we do things like target the ribosomes that are a little bit different in bacterial cells, or target that cell membrane, or sorry, that cell wall that has that different composition in those bacterial cells so that we don't harm the extracellular matrix, for example, of the host animal cell.